Hey everybody, it's Saturday. Today is it July 18th, and this is Science Saturday. Today we're going to talk about the scientific method and what we learned in the textbooks and what is actually the way things work. Should be fun today. My name is Penny Higgins. I'm the founder and operator and all the things for Epic Isotopes, Epic Analysis, which is a geochemical company right out of Wayne County, New York. Our goal is to provide local farms with analytical services rather than having them needing to send things out a long ways. But also, one of the main goals of Epic Analysis is to provide a connection between the general public and science and how it really, really works. So there'll be three different kinds of broadcasts from Epic Analysis. There's Friday Headlines, which I did last night, Science Saturday, which will be about a 1 hour live stream of me just doing science or just rambling or whatever on Saturdays at 1 o'clock Eastern Time. And then there will also be journal clubs. They're coming soon. We'll start out with free stuff, but eventually it'll become a subscriber kind of deal. Eventually, hopefully by this time tomorrow, we'll be able to stream to YouTube. And also, Epic Analysis is looking for more followers on Facebook so that we can actually stream to our Facebook page instead of to Penny's personal page. So, that's the thing. All right. So, anyway, here we are. Uh, Yeah, let's talk about the scientific methods. This is one of those things, right? I'm a scientist. I have a PhD in geology, master's in chemistry. I've done science or wrote a dissertation, many, many publications. And when you're a kid in school, you learn that what's right off to the side here, is how science works. Let's see if I can point the right way. There we go. You start by asking a question, you make some observations, you form a hypothesis, and then you test it and draw conclusions, and then you report results. Well, that would be all fine and good if that's how things really worked. But I will tell you, and I think every scientist will tell you, that the reality is it don't work out that way. But we do, at the end of the day, what is important is that we do form a hypothesis and we test the hypothesis. These things are true, but the rest of this could happen in any order. So I'm going to talk about some research I'm working on right now. I just noticed I am bleeding. That's not so good. We are right now broadcasting live from what I call the spaceship. Right now the lights are on, so it doesn't look so much like a spaceship. But behind me, what you see are actually my lab equipment back here. It's hidden behind this thing. Let me make it go away so you can see. Yep, there's a microscope and a centrifuge and other microscopes and other things. This is where I actually do some real science right here in my house. Okay, anyway, scientific method. So ask a question, make some observations, do some research, develop a hypothesis and test it. And then once you've drawn your conclusion based on your testing, you report the results as a scientific paper. That's all great. Well, let's talk about something that I am actually working on right now, and who knows, we may be doing some thinking together. In fact, as I'm sort of bumbling through all of this, I invite you to comment on the stream your own thoughts. Now, I'll be honest, right now, the way I have this all set up, I can't actually see the stream. So I'm assuming it's there, but I can't see your comments. So I'll probably have to come back later and respond to them. Although I may just, dive in and see what's going on. Okay, so that's all fine and good. So my experience personally, I do a lot of work with studying the chemistry of fossils and rocks to look at past environments. And I've spent some time looking at the Paleocene Eocene thermal maximum, looked at climate change at the last glaciation, and so on. So let's quickly look at some data how we recognize. Oh boy, I'm hidden in here somewhere. Oh, there I am. Let me see if I can move myself so you can see me too. All right. Well, there we go. I moved my camera a little bit so you can see me too. Isn't that cool? All right. So this is a famous diagram by Zachos. And what we're looking at here is a lot of data. All right. I want you to concentrate hmm, on the, what is that? That's the left side. The where it says Delta 18O at the top. That looks pretty fancy. And what we're looking at is the bottom there where the little dots start is when dinosaurs went extinct about 67 million years ago. And then the top is right now. This is kind of in the order in which rocks would be laid on top of each other. And what we're looking at is geochemical markers from ocean cores. And these are from single celled organisms that actually produce shells. They're called foraminiferans. And we can study the chemistry of those using stable isotopes, which is my specialty, and come up with a temperature for the ocean over time. And what you see when you're looking at this graphic at the bottom in the red, it says temperatures in degrees C, and to the right it's warmer, to the left it's cooler. And so what you see is that over time it's gotten cooler. All right, that makes good sense. Okay, so what we're interested in right now is you see in red, right about here, you see the late Paleocene thermal maximum, or it's also called the Paleocene-Eocene thermal maximum. This was an interesting blip as the Earth was getting as warm as it ever was in the last 65 million years, where it got really, really warm for a few 100,000 years, and then it got cold again. Now, this is particularly interesting to scientists because at that time, at the beginning of it, we didn't have any of our hoofed mammals yet. They hadn't arisen, they hadn't evolved yet. We also didn't have primates, but afterwards we did have primates, and we did have the ancestors of horses and rhinos and deer and all of that came about right then in that 200,000 year period. So it's interesting. We also have above that we have the early Eocene climactic optimum. That is when it was as warm as ever. Other things interesting to note right here at the end of the Eocene, the beginning of the Oligocene, that is when glaciers first covered the Earth. So before that, this lower part, the beginning, that first 30 million years after the dinosaurs went extinct, their world was ice free. It was warm. And then after this, we have glaciers. And you can see the um, the line basically is going, it's warm, much warmer, and then it gets cooler. There's a little warm spot there, but it still is ice. And then we're steadily getting colder and colder and colder and colder to now. So we're actually right now in a period of coolness. At least that's where we should be. Now, these are looking at ocean core, so this does not record the most recent 100 years or so. So that's been removed from here. We're not even going to discuss modern climate change, but it's there. It's something we can talk about at a later date. All right, so right about here, near the top is when we have the end of the glaciation and we have the loss of the megafauna. This is when mammoths and mastodons and woolly rhinos and all these interesting things went extinct about 10,000, 15,000 years ago, depending on how you want to do your math. And also humans have been around the last 2 million years or so, so we're pretty recent here, right up here at the top. All right. So that's another episode of interesting climate change because the glaciers receded and it did get warmer and it's pretty complicated. Oh my. Also, just in case you wonder, I run Mew Mew House, which is a retirement home and hospice for cats. This is Sunshine giving me her opinion. I just woke her up. Sorry, Sunshine. Come on. All right. Okay, so some of these have been really tested a lot. We've been investigated the PETM a lot. The Eocene Oligocene transition has been studied a lot. The end of the last glaciation has been studied a lot. But what hasn't really been studied is this right about here, which is the mid Eocene climactic optimum, which is interesting because we're on a trend of cooling, but there's this little blip of warmth. Now, why is this important? First of all, nobody's really looked at it. But secondly, um, we are right now supposed to be in cooling, and we're dealing with a little, maybe a blip of getting warmer as well. And we'd like to know how organisms respond. The other interesting thing here, and then I point this out, this is from Ocean Cores. Well, I don't know about you, but I don't really identify very well with single celled organisms. I don't know. I mean, I know some people who could be single celled organisms, but it's hard to really think about how climate change affects us when we're talking about single celled organisms. Now, the benefit we have here is like with the Paleocene Eocene thermal maximum, we have mammals that are recognizable. Up here, we have lots of mammals that are recognizable because we're talking about horses and bison and cheetahs and things that, wolves that we are very familiar with. Um, but then we have this spot right here, and we'd like to know in this particular environment, we have a little bit of warmth that may or may not have affected um, animals as they were, evolving, living their lives, doing whatever. We do know some interesting things here in the Paleocene Eocene thermal maximum, where it got really warm really fast. Um, it didn't last very long. There was a lot of dwarfism in organisms, but it's tough to really relate that to modern mammals because, first of all, these are things that you probably wouldn't recognize if you saw them. You wouldn't be like, "Oh, that's a sheep. That's a rhino." Um, because there weren't primates in the way we understand primates today. There weren't horses. There were none of those things. They're very different. Whereas by the time you get into the middle of the Eocene, we are talking about things that you'd be like, "That's a rhinoceros." Wasn't a rhinoceros, but you would recognize it as such. And there are some primates that you would be like, "Yeah, okay, that's a monkey-like." There's rodents. There are other hoofed animals running around. Some things are unfamiliar, but we've got larger animals that we can relate to a little bit more. But folks haven't really spent a lot of time and energy studying these things. The other challenge is, of course, that we have to look for the right environment where we have the rocks that have the mammals in them to look at, and there have to be enough of them out there. Now, there's one place in Montana where they've been able to look at this Middle Eocene climatic optimum, which is pretty interesting in a terrestrial record because not only are we not single-celled organisms, we do not live in the ocean. Um, some people like to live near the ocean, but we are not aquatic life forms. We don't. We tend to suffocate underwater. So, so what can we do? So recently, I became involved. Recently, gosh, it's been actually quite a long time. Never mind that. I've been involved with a project to look at the organism changes over time. Um, Mammal changes in North America, so we can learn something about how organisms, mammals in particular, um, deal with warmth. So so here's a interesting another diagram. This red line here, gosh, I don't really have an arrow that I can point at this for you. There should be a way I could do that, but I don't know how to do it. So I'll figure that out eventually. But if you look at the red line that's like right here, um, over time it starts out kind of low and it's moving up. So where it says PETM at the top, when you look at the top here, it says PETM, that's the Paleocene Eocene thermal maximum. The EECO is the warmest period. And you see the red line again, that's temperature. And then you go to the MECO, that's the Middle Eocene climatic optimum. And you can see a little bit of a jump in that box in the red wiggles. And that's the episode of warming. All right. That's all fine and good. So the blue line, on the other hand, is a measure of diversity of mammals. All right. And what you see is as the line goes up, as it goes up, there's more diversity, and as it goes down, there's less diversity. Um, Over time, it has been going up, but then it hits the Miko and it drops, and so we call that the Bridgerian crash. Um, Interesting. So I'm not entirely sure why that is. My name is Penny. I'll be your blonde this evening. All right. So, but what we do see, whatever we see, is that we have an abrupt decrease in diversity in mammals about the time of the Miko. Maybe that's not the Bridgerian crash. Maybe I'm just talking and I don't understand something. It's a possibility. We're interested in this. We want to know more about this. What's going on, and where can we see that? Well, we can go right to the Uinta Basin of Utah, and I'm pulling something out here. There we go. That's beautiful. We can go to the Uinta Basin of Utah. Oh, just close. Go away. And we actually have rock units that expose all of this, starting with the Wasatchian, Bridgerian, Uinta, and Duchanian. Now, what the heck are those? Wow. See, what started out as an easy question, right? And this is the thing of science, right? It started as an easy question. What happened to mammals during this Middle Eocene climactic optimum? Okay, that's great. What happened to mammals? Well, we have to be able to measure mammals, how many mammals there are, diversity. We have this over here. We can look at that. But then we have to also understand how we talk about mammals and naming species. And so, one of the things we are going to talk about in whenever I do it, that it's going to be me talking about the one new species of mammal I named and the process of naming new species of anything. That's something that is very poorly understood by most people. All right. So, what are all of these things along the bottom? Wasatchian, Bridgerian, Uinta. These are North American land mammal ages. And so we can use the different species of mammals present in a rock to put them into these categories. Um, and then once we have them in the categories, we already have exposures of rocks and we know where these categories lie above each other, below each other. So we can use the assemblage of mammals to assign rough ages to things. These are not numeric ages. I have to make that clear. We're just putting things in order. All right. So here's the DU1 is Duchanian 1. And then if you go up, DU1 is where the diversity is lowest. UI3 is Uinta 3. Um, and that's where diversity is the highest. All right. So let's do something crazy. Oh, it worked. Yay. I hope you can see this. This is yeah, go away. It's giving me, there we go. I'll make it a little bit bigger so we can see it just a little bit better. This is an image of a whole slew of data. Wow. And I have to think about this for a minute, because what really happens when we're doing science is that we wind up just throwing out a whole bunch of data and leaning back and thinking about it. And I think that's the important thing that I can't stress enough, is that doing science isn't always knowing exactly what's going to happen. Sometimes it's just collecting data until something makes sense. Yes, I know, Sunshine. You disagree? She apparently disagrees. All right. But what you see in green there is the Miko again. The yellow bar with the blue in it is the Miko. Um, and you can see um, where there's an increase in temperature and so on. The green bar, the green data there in the outline is temperature measured from rocks in Montana. So this is one of the first time we've actually identified the Miko in terrestrial rocks. And so we have it. We actually see it in terrestrial rocks. We also see it in the marine. Down below where it says marine Miko, you can see again, this is the same data or similar data where you see a little peak in the temperature right there, but it's more towards the end of the Miko. And then off to the right, you see a whole bunch of data. Oh, I could do this. Oh, check me out. Um, I don't know if I can actually make this work, but we're going to try it. Um, oh, oh, look at that. See, I can make that go away. Oh, look at that. So one of the things I hope everybody understands is that this is me just exploring, all right, a little bit. All right, so that's carbon and oxygen data. There's the terrestrial Miko. Um, where am I looking at? There's the Menthner stuff. Okay, so that's the terrestrial Miko. Okay, so we're looking at that. Now, what we already had from Central Utah was all of this data, which is all on the right-hand side of paleosols and lake deposits, and also tooth enamel samples. A lot of these things have been unpublished. But when you compare that to some of the other temperature data, it seems to follow a general trend of getting cooler. And then we have this little blip of warmth right there, which is the Miko. All right, so we have all of this data. And here is the shocking thing about how science works. I'm going to go back to this. The shocking thing about how science works is that a lot of times what happens is that we just kind of lean back, shut our eyes, and think about what we've got. We plot the data, we look at it, scratch our heads, take a shower, and then we get ideas. Oh, my lights just flickered. That wasn't so good. There's a big old thunderstorm going on outside, so let's hope that the power doesn't die. Just so y'all know. So we asked the question, it seemed like a simple question, well, what happened to mammals during this little global warming? And we've got all of these observations. We know there was a dip in diversity, and we've got some isotopic evidence that should show us where it is. And we have to now develop a hypothesis. All right, so what is our hypothesis? Well, the hypothesis would be, that the Miko caused a reduction in biodiversity. All right, that's the hypothesis. Now, is that true? Well, we have to ask ourselves, first of all, we have to find a place that actually has all of these organisms where the Miko would be, and then localities where we can look at the mammals. And then we have to convince ourselves that we know where the Miko is in these rock layers, which is where I come in. And then we have to conclude things. Now, some of the hypotheses, some of the things we have to test, again, is knowing where the Miko is. We also have to convince ourselves that diversity actually did decrease, and it wasn't just because we had a sampling problem. What do I mean by that? So, just because we see a decrease in diversity, that could be because where we've collected mammals, we just haven't collected the right localities. We have to think about how the fossil record actually works. There aren't fossils everywhere. They're not the same distribution everywhere. Different environments had different organisms. And when we look at the rocks themselves, let's look at this again. Um, where's the Wasatchian down here? Let's see if I can find this again. All right, the Wasatchian here. Well, that's actually lake beds when we're looking at the terrestrial record. Over here, the Bridgerian, okay, and the Uinta, this is a river area. There's a lot of soil development. And then in the Duchenian, it appears to be a little bit wetter, and we don't really have heavy duty soil development. And we have to ask ourselves, this is all continuous record, all on top of each other, but you're not going to find mammals in lake beds, at least not the mammals you're interested in. You might find them up here. The environment of deposition in the Uinta may be just perfect for preserving mammals. And as it happens in the Uinta Formation, in the Uinta Basin, we do get all the really cool things, the Archaeotherium and the Brontotheres and then tons and tons of turtles and garfish, okay, which is like, wait, what? And then you move up into the Duchanian, and the rock environment is completely different. The rock color is different. Um, the environment is different. There's a lot more rivers, there's less soil development. So maybe it's not because there are fewer mammals. Maybe it's just the environment wasn't really conducive to preserving them. So we have to assess that. So we have to develop a test to assess that. And then we have to develop some tests to decide if we can actually put our finger on the Miko. Okay. So I'm going to come back to this. And what we have going on in the Uinta Basin, I'm going to make this go away if I can make it. There we go. What we have in the Uinta Basin is we have all of these rock records. It's raining really hard right now. We have all of these rock records, and we see cooling in oxygen data and everything, maybe. But we actually don't have rocks or we don't have data when we started this that was where we predicted the Miko to be. So we had to go in and do some different work. But also, when you look at the bottom of this diagram, you can see there's the Uinta Formation and the Duchesne River Formation. And notice that the Duchesne River Formation is where the Miko should be. And also, as it happens, as I said, the Duchesne River Formation might not be really conducive to preserving mammals. So we're working on that. But also, we need geochemical data to prove to ourselves that the Miko actually is there. And that's the thing that I've been working on for quite a while now. So, again, when we look at this thing, we have really great fossils and geochemical evidence all the way up into UI3, but we don't have much from the Duchanian. So we've got some work to do. And that's what we've been doing. Okay. Where am I at? Okay. So we've done us, we're a big project, a lot of people covering all the time zones all the way across the United States. We've brought in people from Canada, we've worked with people overseas trying to understand what's going on. Forgive me while I have a drink. Now I just realized that I need to add an image here. This is all right. As of well, that was cool. I don't know what I just did. Oh, I didn't want to do that. Data as of cancel. What? I just did that. Okay. Let's try this again. Image data as of. All right. I have something weird going on here. All right. Image. Okay. Let's just do this. It's doing some weird stuff. I want to go here. Oh, hi. Yes. No, no, you may not. You may not jump on this. Yes, I know. Okay, let's put that there. All right. So check it out. We do have data. Oh boy. Yeah, but this is a mess. Holy crap. All of these little dots that you see are all individual data points. Holy crap. And this is all from the Duchesne River Formation. This is our latest work, and I'm leaning back, scratching, and I did all this work and am now at the point of staring at it. Now the pink dots, those are color data, And in that particular case, that's just magenta of all things. So we are able to measure the color of the rock using some special light. Oh, let's think about it. Spectral analysis. There we go. We're doing that. So you can measure L star, A star, and B star, which are common measurements. You can also measure magenta, cyan, and yellow. And I work at Kodak, so I have a very special place in my heart for magenta, cyan, and yellow. Um, you can look at red, green, blue, and all of that stuff. And I just picked magenta because it seems to be the only thing that shows any kind of pattern, and it really doesn't seem to show much of anything other than being flat. And maybe having a few little episodes of kind of going negative, like right there and right there and over here. Okay, so it seems to get lower. I'm not making any sense of that. The green line is a measure of grain size. This is the largest measurement of the largest clasts found in the rocks. And it's kind of reversed from what you think. So um, lower is like coarser grain. So these are like cobbles and gravel, and higher is like silt and the really fine grain stuff. And what you see is like right here, it says coarser, and that's actually a conglomerate. So we have conglomerates in this area right here. These letters at the top, there we go. The letters at the top are the individual sections that we measured. And then we have the more interesting stuff here. Up here, this red to blue line, this is the geochemistry. This is the isotopic analysis of various rocks and soils throughout the section. And we're able to use those isotopes to calculate mean annual temperature. And we're not going to talk about what the actual temperature is because there's so many variables. But what we are going to say is that up here, when it's higher, it's warmer, and when it's lower, it's cooler. And so if the Middle Eocene climatic optimum is a warmer spot, we're looking at something that's higher. But what we have, interestingly, is this much lower spot near where we also have the coarser grain stuff, which is curious. The other thing we have going on right here, and this is this green to brown line right here. This is using a different spectroscopic method called X-ray fluorescence, and we're looking at the percentages of various oxides in the soils and the paleosols in particular. And you can use these things to calculate mean annual precipitation. And what you see is that for the most part, it's wet-ish, but there's a dry spot right there. So here we go. We're like, okay, so if it's the Nico and it's warmer, all right, that's fine. Then we got this cooler spot. We also have this drier spot. And then you have to ask yourself, this is one thing when you're in the ocean, like the whole ocean reflects the whole world. And when you're dealing with land, it's way more complicated than that because not only do you have, it's not averaging one area, it's the whole world is averaging that area. It's the immediate environment. And so dry spells, warm spells, how many millions of years is this? We're not sure yet. Is it warmer? Is it cooler? It could be. I mean, for example, when we look at like Europe or like, what am I thinking of? Yeah, Europe in general is much warmer than North America at the same latitude. Um, and that's because they get all the warm water from the Gulf Stream. Is that right? Yeah. So, we can't really say, and I don't like to assign values to these temperatures because they don't necessarily make much sense. So we're thinking about relative changes. We also have to add to this the complication that, as it happens, there was a mountain range coming up right next door, like within 50 miles. There's a mountain range that was coming up at the same time. Now, mountain ranges tend to affect climate. So Are we looking at temperature changes because of global change, or are we looking at temperature changes because there's a mountain range? Are we looking at it getting drier because the mountain was blocking rain from getting to this area, or maybe it changed the flow of rivers? We don't know. So I guess my point with all of this is, first of all, we've hit the point where we've plotted all this data, and we're going to lean back, we're going to look at it and think about it for a while. We do have some other data that are going to get put on here, including some geochemical, not geochemical, but it is geochemical, but we have some radiometric dates we're going to put on here. um there's a doctoral dissertation out there where somebody looked through here and examined in the conglomerates and in the pebbles and whatnot, what part of the mountain range that was coming up was actually eroding into this space and at the time. So we can actually compare mountain uplift with climate change, which would be kind of cool. But I got to sort through all that. Um, and this right now, I haven't put this against any of the Uinta Formation data yet. That same data that we were just looking at right here. Okay, so all that stuff on the right hand side of this diagram hasn't been set next to all of this new data that we're looking at right now. So we got to see how this is going to all go together. I have to do some math, I have to do some other things. We need to make sure we got everything correct. There's a ton of work here. So my point being with all of this is that, let me see if I can do this. No matter how easy of a problem it seems to have been, testing the hypothesis winds up being really complicated. And this is kind of how science really works. Um, a lot of this is me plotting data, sending it off to my colleagues and saying, do you see what I see? And we question each other and we smile and nod. Um, and then we ask ourselves, what our hypothesis is, and are we actually testing what we think we're testing? Are we asking the right questions? Do we need to change the way we ask our questions? Um, and the last bit of this where it says, let me make some of these things go away now. Where it says, use this hand, draw a conclusion, report results. Okay, we're still in the middle of testing our hypothesis, but we've learned a lot. We've learned a ton. We're not quite certain yet that we found the Miko in the Duchesne River Formation, but we're pretty confident that it's there. And it just looks different in a terrestrial environment than it does in the ocean. And so we have to tease apart some of these things. But we can still answer some questions. First of all, we can go through and we've walked those sections and we've looked for fossil localities. We've looked all over the place. We have some, and we're looking at the actual diversity. Okay, so we have to know if diversity is actually dropping, and other folks are doing that work right now. We're trying to understand that. We're going to get there. Because we can't really be sure. And then the next part is we've got to put all of this data together. So probably what's going to happen is when we get to the draw conclusion point here, there's going to be a publication that comes out that's going to be nothing more than saying, we found the Miko. We found it in the Duchesne River Formation. We have to convince ourselves of that. And how we know it. It might not even have anything in it about the mammals. It might not have much in it about some of the other things we want to know. It's not going to be actually answering our initial question, which is, did the Miko cause a real drop in diversity? What we're answering instead is, well, we found the Miko in terrestrial environment. Hooray! And that's how science really works. When you're going through it, it's never, ask a question, develop a hypothesis, test the hypothesis, and then you're done. It's ask a question, make some observations. Sometimes you make the observations first. That's happened before. You make some observations, you develop a hypothesis, and in the process, you come up with more questions. And before you know it, a simple question becomes a lifelong research project. And that, my friends, is how science really works. And what I've just gone through with you is what I'm doing right now, and hopefully it's interesting to you. And like I said, um, I don't exactly have access to any comments or anything right now. But if you have any thoughts about this or suggestions or things that we could do, then by all means, please put them in the comments. I'll follow up because, what I've learned, this is the most important thing. If you take nothing else from this is what I want you to know the most. You don't have to have a degree in science or any special training to be helpful here. What I've learned in my many years of experience doing science is by taking data like this and just sort of telling people who may or may not know anything about it what's going on. It's the person that doesn't have the bias, the preconceived notions, the training that says, "Oh, you can't do that." They're the ones who notice the patterns that I miss every single time. When I would do field work, I'd bring students out with me, and I would send them places where, you know, I'd be like, I don't know, there should be something. My best example is we're standing in the field, and this is looking at the Paleocene Eocene thermal maximum. We're standing in the field, and I'm like, guys, I know there is a fossil locality around here somewhere, but I have walked everywhere. I have never found it. It has to exist, but I haven't found it. Because in my head, I knew what I was looking for, and I just didn't see it. So I said to my students, I said, go look for this fossil locality. It's out here somewhere. Just go find it. Don't go so far that I can't see you anymore, but just go look for it. Took them about 40 minutes. They came back and said, yeah, we found it. Took me to an outcrop that I had never even paused at before because it did not look like the right rock, and I had already decided in my head that that was not the place to look. And there it was. And there were all these fossils, and these were fossil clams, but I was like, yep, yeah, that's it. So if you have thoughts, the moral of the story is that if you have thoughts based on what I've told you today, or over the course of, however long I continue to do this, um, if you have thoughts, ideas, suggestions, questions, then ask them. Put them in the comments. Share with me. We'll discuss them. And when I get to a place where I'm doing the journal club, then we'll talk about them there too in more detail. If you have thoughts about things that are happening in the world today that maybe, are related to science, geoscience, then ask them. Don't ever be afraid. Science is everywhere. Science is among us. Science isn't just, scientists on one side, a wall, and then the rest of the world. We're all scientists here. Some of us have a different kind of training than others, but we're all observers. We all see things. We're pattern seekers. And if you see something that I've missed, then please tell me. All right, everyone, I'm going to go on ahead and step away from this because I can. Wow, and the thunder's getting a little bit louder, so maybe I should do that. I encourage you to please follow Epic Analysis on Facebook. Yes, right now it's Epic Isotopes, which is why I kind of said Epic Isotopes. I will eventually change the name, but if you follow me there, I need 100 followers so that I can live stream there. I will also take a recording of this broadcast, and it will wind up over on Epic Isotopes, and also I'm working on a YouTube channel so that I can stream over there as well. I want to hear from you. Ask me questions. Tell me what you want me to talk about. And yeah. And if you're feeling good, if you like what I'm doing, then keep following me, share what I'm talking about. I also, as some of you know, run Mew Mew House, which is a retirement home and hospice for cats, which is why there's a cat sleeping behind me right now. That's Sunshine. If you feel like it, you can also support me indirectly by supporting Mew Mew House. Part of the reason why I launched Epic Analysis was so that I could make some money so that I can feed the cats. So if you feel so inclined, you can go to mewmewhouse.org, or find Mew Mew House on Facebook, and you're more than welcome to support me in that way. That money goes straight to the cats, and honestly, any money I make here is probably also going to go straight to the cats. So, skip the middleman. All right, everyone. I see that there are a lot of thingies, notifications on Facebook. I haven't looked at them yet. So if you have any thoughts, questions, comments, reach out. Again, my name is Penny Higgins. I have a PhD in geology and master's in chemistry. I'm a geochemist. I'm a paleontologist. And soon, now, I'm bringing the agricultural lab right to your backyard. I hope everybody has a wonderful and fantastic rest of your weekend, and I will see you next Saturday, 1 o'clock Eastern, for Science Saturday. Bye-bye now.